那谢谢大家来参加我们这一场的那个科技新知研讨会。那我先大概用中文介绍1-2分钟，然后我们就用英文introduce我们今天的从日本来的讲者。好，那这一场研讨会是这个科技新知研讨会是由国立台湾大学跟国立成功大学其实一起来办。那国立台湾大学除了我们台大的生技中心，那我个人是现在担任台大生技中心的主任，我叫沈汤龙，叫沈汤龙。那另外我们国立台湾大学也有台大医学院跟台大医学院那一边的一些像药品创新创业的一个推动中心一起来去合办。那成功大学是我们这边汤明哲教授他之前成大的教务，然后他现在也担任国立成功大学伤口恢复与再生中心的主任。就是由政府的生根计划所支持的一个持续支持的一个中心，希望把很多相关的基础研究把它推展到一些产业化的东西。那今天这个演讲，我们特别邀请了从日本国家癌症研究中心的松崎润太郎，Dr. 松崎润太郎。那事实上是利用所谓的液态检体，就是血液来去做癌症的一个检测。那大家听到非常非常丰富的一个研究的一个结果。那 So the Juntaro actually is from the National Cancer Center and from the lab of Dr. Ochiya. And actually Dr. Ochiya, he's the president of the Japan Society for Extracellular Vesicles. OK， 就是胞外体。那我自己本身也担任台湾胞外体协会的理事长。So extracellular vesicle, such as the exosome, actually recent year become more popular. And so Dr. Ochiya's lab working on a lot of exosome. But when you want to do the biomarker, so sometimes they also include a free molecule in the blood. So today, Juntaro is going to introduce us the project. He is the key person in Dr. Ochiya's lab. From the AMAC actually is the founding agents， 就像一个跨部会的一个单位，在日本跨经济部、跨他的教育部这些等等。So the AMAC fund their lab for 5 years grant to study or to develop the biomarker from the blood for the cancer early detection. So they got a 5 years grant. Total amount is $80 million $8,000 万美金，5年计划 since 2014. So this year they will end the project, but now all the discovery already on the clinical trial. So in the near future, we believe there's some clinical application will happen, right? At least in Japan, but we are trying to have very close collaboration with the Ochiya and also the Juntaro, and they have very good experience. And Taiwan have very good clinical sample, and we can really learn a lot from them, and to develop our own biomarkers. All right. So today, so you can see the title Juntaro is going to present is the microRNA, but it's from the blood. So we call it circulating microRNA, and then use their own system to develop the profiling of the microRNA for the cancer diagnosis. So not only the early detection, but probably also the companion diagnosis as well, particularly for the immunotherapy response. They also try to work on that. So so the last part actually is Juntaro actually got his MD in the Keio University School of Medicine. So he's an expert in the GI. But he really liked research, the basic research, so he joined Dr. Ochiya's lab in 2015, right? 14 or 15, started working on this AMAC project. So he also got his PhD in 2013, also in the Keio University. So let's welcome Juntaro.
Good afternoon. So, my name is Juntaro Matsuzaki from Tokyo, and thank you for inviting me this great meeting. especially for the Dr. Tang Shen and Dr. Tang. And this is great pleasure for me to share my recent result here. So, the Dr. Tang Shen also said that I'm a gastroenterologist, GI clinician, but now I'm working as a researcher in National Cancer Center. And mainly I'm involved in this project as a statistician, not so kind of molecular science in this project. So this topic I'd like to introduce is mainly about the statistics. So, anyway, I'd like to start. So this is my COI information. So today I'd like to introduce our kind of national project, as mentioned, Dr. Tang Shen. We were supported by the AMET. This is the kind of Japanese NIH, and this is the final year of this 5-year project. So in this project, we focused on the circulating microRNA, specifically serum microRNA. And our final goal is to develop the 13 the kind of blood test for the detection of 13 kinds of different solid cancers. So recently the liquid biopsy area is quite expanding. So this technique also could assist this field, I think. So as a GI clinician, I'd like to introduce a little bit about the current condition in the cancer screening, about this kind of gastric or colorectal cancer. As you know, we usually use the endoscopy for the gastric cancer or colorectal cancer. But we previously did the kind of questionnaires in the Asian countries for the expert GI clinicians, and we found that even in the quite low risk population, Japanese GI clinicians recommend every 2-year endoscopy, and for the high risk population, we recommend every year endoscopy. And in case of the colorectal cancer, our GI clinician recommend every 3 or 4 years colonoscopy, even for the quite low risk population. And for the high risk population, we recommend every 2-year colonoscopy. So this is quite invasive examination. Everyone doesn't like that. So the examination, the rate for the examination is quite lower in Japan, and this examination is time consuming and it takes a lot of human resources. So we need to change this current situation. So in worldwide, so far more than 1,000 of companies are developing liquid biopsy products like this. And in cancer biomarker field, they are trying to develop not only the early stage detection, but they also try to molecular diagnosis and treatment selection and monitoring for the cancer patient. So as I mentioned here, the really exponentially enlarging area, this field. So I'd like to explain a little bit about the microRNAs. So in microRNAs microRNAs are quite short non-coding RNAs consisting of 70 to 25 nucleotides. And these microRNAs can usually bind to 3' nuclear region of messenger RNAs and inhibit the translation of these target messenger RNAs. In this way, the one microRNA can regulate various numbers of messenger RNAs, and this microRNA is known as a major player, quite important player in cancer development and cancer progression. So, and importantly, these microRNAs can be released from the cells like this via the encapsulated in the exosomes or binding to some proteins or lipids. And these released microRNAs can play a role like cell-to-cell communication. So let me explain about a little bit about my PhD student study. So previously I focused on this disease, esophageal adenocarcinoma. So this cancer is a kind of the gastroesophageal reflux disease. And this is the early stage esophageal adenocarcinoma. And we know that intestinal metaplasia of esophagus is the kind of precursor region of these cancers. And this cancer is now in Western country, the prevalence of this cancer is rapidly increasing due to the obesity and decreased prevalence of H. pylori infection. So now we collected the tissue samples of these two conditions, and we found the microRNA 221 and 22 were increased during the development of these cancers as well as the progression of these cancers. And after that, one Germany group reported that the elevated levels of serum microRNA 222 were associated with the poor progression of prognosis of these cancers. So in this time, I really interested in the serum or plasma circulating microRNAs, and as a quite promising biomarker, next generation biomarker. And the important thing is that the microRNAs, especially for the microRNAs encapsulated in the exosomes, have physiological roles, not just released from the cells like garbage. It has quite important role for cancers. And so, as you can see here, the cancer cell can release microRNA containing exosomes, and these exosomes can be taken up to the surrounding cells, and these microRNAs can change the phenotype of the surrounding cells. Like promote angiogenesis or something, and this effect can assist the cancer progression or metastasis. So, for example, we previously reported that some quite highly malignant, highly metastatic breast cancer cells can release the microRNA 181C containing exosomes, and this microRNA can break down the blood-brain barrier. When we inject the quite highly malignant exosome into the mice, the blood-brain barrier in mice were destroyed like this. So it means that the highly metastatic breast cancer cells release these containing exosomes, and we found that the exosomes were taken up by the endothelial cells and change the actin dynamics and break down the blood-brain barrier. And interestingly, we also found that in patients, as exosomal microRNA 181C levels were higher in breast cancer patients with brain metastasis. So it means that this like functional microRNAs can be useful as a biomarker. So in our project, we collected serum samples and clinical information from the various institutes, and we also collaborate with several companies. And in our lab, we did the microarray analysis for all the serum samples collected here, and did the statistical analysis. And we tried to find a molecular biomarker, microRNA biomarker for the detection of cancers. So our cancer samples were mainly obtained from the National Cancer Center Biobank in Tokyo. So in this biobank, we have already collected more than 740,000 serum samples, and among them we used 70,000 cancer patient serum samples, and we analyzed the microRNA using these serum samples. So we used this microarray system provided by Toray. This microarray tip has quite unique structure like this, and this chip is made of the black region instead of the black glass, and this chip has quite unique this like 3D structures, and to stabilize the spot morphology, and this like white beads agitation can enhance the signal intensity using this quite unique microarray system. So we did the microarray analysis using this microarray platform for these numbers of cancer samples as well as non-cancer controls. As shown here, we include 13 kinds of solid cancers. Which includes the quite rare cancers like sarcoma or ovarian cancer. But thanks to the biobank, we can collect quite lots of samples during quite limited interval, like 5 years. So to do such kind of large scale microarray analysis, it is quite important to standardize the protocol. In our protocol, we use the 300 microliters of serum samples, and our total RNAs are extracted from this serum, and we concentrate the RNA templates to 4 microliters, and among these 4 microliters, we used 2 microliters for the microarray analysis. And using this platform, we can handle around 2,500 serum samples per month. And we can obtain the 2,588 microarray expression levels by one microarray chip. And we also identified kind of internal control microRNAs. We selected these 3 internal control microRNAs for the standardization, normalization of the data. And after the selection of highly expressing microRNA in circulation, around 300 to 500 microRNAs were selected and analyzed in the further analysis. And samples are randomly divided into the training and validation set and constructed the discriminant model in the training set, and the diagnostic accuracy was checked in the validation set. So this is the kind of complicated slide, but this is quite easy, quite kind of conventional discrimination and strategy. So first, I selected the best 20 microRNAs for the two-class discrimination like this. And next we combine the other microRNAs one by one like this. And some of the combination can improve the diagnostic performance like this. So, and using the leave one out cross validation, we further selected the best 20 microRNAs combinations. And next, we combine the third microRNAs like one by one like this and repeat it to optimize the diagnostic model. So here's an example about the detection model of esophageal squamous cell carcinoma. In this analysis, we included 566 ESCC patient samples, and we also analyzed more than 4,900 non-cancer samples, which includes our lung samples. And the half of the ESCC samples are allocated in the training set, and the other half samples are allocated in the validation set. And the same number of non-cancer samples are included in the training set. The other samples are included in the validation set. So in the training set, as you can see here, just one microRNA can achieve the diagnostic accuracy with the AUC of 0.9, but the combination of microRNAs can improve the accuracy like this. And we selected the 6 microRNA combination model, which is the optimized levels of accuracy. So these are the 6 microRNAs here for the ESCC detection. So the first pivot microRNA is the best one, and the other 5 microRNAs can assist the accuracy of this microRNA, and the combination of 6 microRNA can achieve the almost perfect discrimination between the ESCC samples and non-cancer samples. And this diagnostic model was nicely validated in the independent validation set. As you can see here, the specificity were more than 90%, 95% irrespective of the institution. So, and we checked each stage of ESCCs, and as you can see here, even in the stage 0, which means the tumor in situ, even in the stage 0, the more than 98% sensitivity can be achieved. So it means that circulating microRNA profile is quite useful for the early stage cancer detection. We obtained such kind of proof of concept here. So this is the another case about hepatocellular carcinoma. In this case, HCC is developed from the hepatitis or liver cirrhosis patient. So it is important that we need to construct the model to discriminate not only non-cancer samples but also the hepatitis or liver cirrhosis samples. And using the 8 microRNAs, we can achieve the quite highly accurate diagnostic model. So this is the case of sarcoma. This cancer is quite rare cancer, so it is not necessary to develop the kind of first screening tool for this kind of rare cancer. But here we try to diagnose the malignant or benign bone and soft tissue tumors using microRNAs. And as you can see here, the 7 microRNA panel can discriminate the malignant and benign bones and soft tissue tumors. And interestingly, when we check the expression profiles of intermediate bone and soft tissue tumors, the diagnostic index level were also intermediate between the malignant and benign samples like this. And when we check the histological subtypes in the intermediate group, there are 3 subgroups show the high diagnostic index. And interestingly, we know clinically these 3 histological subtypes have malignant transformation potential. So it means that the microRNA profile can be changed even before the even in the kind of pre-malignant tumors. So this data also shows that circulating microRNAs is quite useful for the early stage cancer detection. And this data shows the result about the prostate cancer. As you know, in case of prostate cancer, we have already used available a PSA biomarker is available for this cancer. But as you know, this marker have really high false positive rate. So there are lots of unnecessary prostate biopsy. So in this trial, we set the negative prostate biopsy patient as a non-cancer control. Almost all of them were performed needle prostate biopsy due to the high levels of PSA. So in comparing to these 2 groups, the PSA doesn't work as a discriminant, but these 2 microRNA combination can achieve the discrimination of these 2 groups. So, but this case shows the different results. So when we constructed the detection model for the ovarian cancer, we made 10 microRNA models, and actually this model can achieve the discrimination between the ovarian cancer and non-cancer samples. But using this model, we cannot discriminate the borderline or benign ovarian tumors. So this model can be useful as a first screening of the ovarian tumor, but using the microRNA profile, we cannot diagnose the cancer or the benign tumors. So it means that the microRNA profile is not only changed by the presence of cancer, but some types of benign or other tumors or disease can also change the microRNA profiles. The same situation was occurred in the brain tumors. So as you can see here, the glioma and non-cancer patients were nicely discriminated like this. But using this model, we cannot discriminate the glioma and the other brain tumors. So we try to make the other model to discriminate these three brain tumors. It means the glioblastoma and the CNS lymphoma and the metastasis from the other tumors, other cancers. And here we use the logistic lasso algorithm, and we included 40 microRNAs in this discrimination model. And in the training set, we can achieve the comparatively nice discrimination, more than 70% accuracy. But in the validation set results shows the marginally the result. So it means the diagnostic accuracy in the glioblastoma and the brain metastasis was more than 80%. So it would work as a discriminant between these kinds of the other kinds of brain tumors. So next, I'd like to try the prediction of cancer localization. So now some quite high impact paper shown the possibility that just blood test can predict the cancer location. So in this paper used the combination of circulating tumor DNA and protein panels, and they mentioned that circulating tumor DNA cannot predict the location, but some protein markers can estimate the cancer location. On the other hand, the other paper focused on the methylation status of cell-free DNA, and they succeeded in estimating the cancer location using the methylomes. And we also tried this point because we know the tissue microRNA profiles are quite distinct among the cancer location, like this. So we assume that the circulating microRNA can be the most accurate biomarkers for the prediction of location. So in this case, we use the deep learning system like this. So as you know, the recent progress of the image recognition like this is quite improving in this couple of years due to the introduction of deep learning algorithm. This is really good at the image recognition, and analyzing the microRNA data is quite similar to the image recognition. So we try to adapt this algorithm to the microarray analysis. So we fed the data to the deep learning system, and actually we are quite surprised that this deep learning algorithm can distinguish these 13 kinds of cancers and non-cancer samples quite accurately like this. So this actually in this analysis we used all 2,500 microRNA profiles for this analysis, but we also obtained the proof of concept that microRNA profile can estimate the tumor localization. And importantly, using the machine learning system, we can calculate this kind of contribution score for the 14-class discrimination. I mean, some quite famous microRNAs were ranked in the high score, like this. So this quite high score microRNA is quite important for the discrimination of cancer location. So now we are trying to clarify the function of these important circulating microRNAs. And we also focused on not only the cancer detection, but microRNA profile can also be useful for the other situation, like the prediction of lymph node metastasis like this. In breast cancer, for the detection of axillary lymph node metastasis, now we use the sentinel lymph node biopsy procedure, but this biopsy still has some invasiveness, so we try to predict the lymph node metastasis before the operation. And we found 2 microRNAs were statistically different among the lymph node metastasis positive and negative group, and the combination of these 2 microRNAs can enrich the lymph node positive group. And we also combine the microRNAs and clinical information, and the prediction of lymph node metastasis was quite nice level, like AUC of 0.05, 0.85. So I'd like to introduce the finally about the immunocheckpoint inhibitor. So this is quite small sample size trial, but quite kind of pilot study. But you know the immunocheckpoint inhibitor, like anti-PD-1 antibody. In this case, we use the nivolumab. And this drug is quite expensive, and the response rate is less than 20%. So it is important to identify the responders for these drugs. So we collected blood samples from the esophageal squamous cell carcinoma patient before the treatment of nivolumab, as well as the 2 weeks after the treatment. And 1 microRNAs were significantly associated with the treatment response. These greens are responder, green and blues are responder. And 3 microRNAs were likely associated with the treatment response in the blood obtained 2 weeks after the treatment. So like this, circulating microRNAs also be useful for the identification of responder in some treatment. And finally, we also try to adapt the other concept than the cancers. So this case shows the stroke. So we found we collected the samples from the kind of silent infarction, like the kind of high-risk patient for the stroke. And in this kind of high-risk patient, the circulating microRNA profiles were a little bit different from the healthy population. So we also constructed 3 microRNA discrimination model from these 2 groups, and these kinds of microRNA can be useful for the risk prediction of stroke, and maybe we can do some integration for the prevention of the stroke. So in summary, circulating microRNA profile provides us quite accurate cancer diagnosis quite in early stage, and it also predicts cancer localization, and it also can be useful to improve the therapeutic strategy. And so now we developed this kind of huge database. It includes more than 50,000 serum samples data, and but in this data we mainly use the biobank samples. So we need to check the data using the freshly collected samples, and then we are collaborating with some companies and try to approve try to obtain approval as a clinical application in next few years. And we also find the importance of the investigation of the molecular mechanism of circuit microRNAs. You know that we have huge data which is statistically significant in terms of the cancer detection, but we don't have the reason why these microRNAs can discriminate cancers. So in this case, if we do not know the molecular mechanism, we cannot assume the reason of false positive or false negative in the clinical application. So this is quite a problem. So now we think that the circulating microRNA profile can be changed not just due to the presence of cancers, but as I mentioned, the case of ovarian cancer, the benign or some other disease also affect the circulating microRNA profiles. So we need to know the main player as a donor of circulating microRNAs, and we further investigate the reason why the presence of cancer can affect the circulating microRNA profile. And the other important thing is that we should know the physiological function of these circulating microRNAs. These microRNAs can be taken up by some cells, some sort of cells, and it have some function. So if we can know these kinds of mechanisms, this circulating microRNA can be a therapeutic target in some diseases. So finally, I'd like to acknowledge my lab members and biobank and clinical collaborators in hospitals and several companies and AMED. So thank you for listening. I'd like to take any question.
It's a very wonderful talk, and I guess you probably can sense there's a lot of study that's going on, and the progress of the biomarker, particularly from liquid biopsy. And today we are trying to invite Juntaro here because he's young, actually he's in his 30s. Okay, so Taiwan and Japan is very close. So I really encourage all of the young scientists or students, you really, use Juntaro as a role model and try to pursue your research and do the collaboration, international collaboration, particularly Taiwan and Japan is very close by. And so actually for me, I know Dr. Ochiya just 3 years ago, but we already back and forth with each other more than probably across the 10 times. And so we are very good friends. All right. And Juntaro actually is the key person, so he helped us a lot to doing all of the database for us. Okay, so, and we have to really break our somehow 成见. So in Japan now they are very, very open. So the cooperation actually is not only in Japan but also overseas. Taiwan, Korea, China, Australia, US. So particularly like the Otasens's companies, AI company actually in San Francisco. So we can really use this whole networking and to help your research. And Juntaro just joined Ochiya probably 5 years ago. He already published more than 70 papers. Okay, so using as a young scientist, you can reach to so many papers or publications in very, very short period of time, and you should learn from him. Okay, he is from the clinician and shift to the basic research, although he still have to do some practice as well each one day per week, but he still spend a lot of time in lab. So try to talk to him if you have time. Okay, so now we open for questions if you have any. Okay, student first. Okay. Any?
Okay, well, I'm not a student, but right.
Just speak up.
Okay. You talk about using the similar to image recognition system as your like AI-based model. Would you please elaborate a little bit more? So specifically, did you use the microarray data like the image for this recognition, or you actually calculated? Because I'm trying to see whether you can transfer. If you use like number-based, then you will be able to use actually just sequencing data. You can also convert it. So would you please explain?
Thank you for your great question. So, as you know, we can obtain the data, not just the image. We can obtain the data from microarray analysis. So, in this case, the image recognition and the microarray data is kind of analogy, but actually we use the data itself for the AI for the deep learning system. So it is also would be useful for the data of NGS or other materials modalities. Yeah.
So any other questions? Yeah, just anything you cannot catch, you also can ask. Okay, try to really speak out in the audience. Okay.
Thank you very much, Dr. Juntaro, for your very informative talk. I'm interested in the sampling methods that you use to collect the serum samples you used. So there are two questions. First, is there a standard protocol for taking these samples? For example, it has to be venous blood, fasting, or postprandial. And secondly, do you think that the way of sampling will have an influence on the circulating mRNA profile? Thank you very much.
Thank you. Yes, it is a really important question. So initially, we think the circulating microRNA is quite stable. So the sample handling would not so much affect the quality of data. But actually, during this project, we found that yes, circulating microRNA and serum microRNA can degrade during the preserved periods, and it can be affected by the sample handling. So, actually, in the biobank samples, it is of course the preserved interval is different between samples. So we overcome this limitation by the sample size. But in case of prospective validation study, we use the freshly collected samples. We made quite strict SOP to collect samples. But actually, we need to obtain the samples from cancer patients. So we don't standardize like fasting or we don't standardize the patient condition. But after obtaining the samples, we handle very strictly So actually we can share the protocol for the freshly collected samples. So if we try to compare the data each other, it is really important to excite.
Thank you very much.
So any other questions?
I have a question. One question is what is the expression level of each microRNA you select in this microRNA profile? The expression level.
So you mean the kind of fold change or something?
Yeah, compared to control, how many folds
For
you
change, for
select
change.
in this profile.
Yeah. Yeah, actually, yeah, I showed the in case of the esophageal or squamous cell carcinoma cases. Yeah, as you can see here, this kind of really important microRNA to discriminate two groups. The fold change is around 2 or much more. But the other microRNAs, as you can see here, the fold change is not so high. So it means that these microRNAs were not specifically introduced in cancer patients, but most of these microRNAs were also exist in the non-cancer controls and a little bit higher or lower in cancer patients. And we need to combine these changes and make the discrimination model. So it means that the detection, so the technology for so the important thing is that the technological error would be really small, should be really small, because if the data replication is poor, this kind of small change cannot be detected. So the technology should improve much more, I think. So
yeah, the fold change probably will need more than two fold change.
Okay.
Yeah, but then combine the different microRNA together, so they will give a better accuracy.
Because I want to know how can we select the microRNA to put in the profile to predict space the cancer or the disease. So how can we so have any different step? One whether the first step is the expression level or the first step is based on the clinical the correlation.
So how you select the featured microRNA before do the modeling? How those microRNA will you select before the subject for the modeling for the index? So you have 300. How you select the 300?
yeah, so we selected microRNAs not only the fold change, but the important thing is that the discrimination accuracy. So as you can see here, even the fold change is not so large, not so big, the SD, if the data of the SD is small, these two groups can discriminate accurately. So we aggressively include this kind of microRNAs to improve the discrimination of two groups.
So all depend on the statistic analysis. It's no phenotype yet.
Whether this microRNA you select is located in the extracellular vesicle or not, just in tubes.
Yeah, in this case, we use the total serum microRNAs, not only the exosomal microRNAs. Actually, initially we tried to do the exosomal microRNA. But when we consider the handling of this kind of huge amount of samples, it is really bit complicated procedure. So first we try to do using serum microRNAs.
Third question, whether this microRNA can target a specific gene or not, have a similar target gene.
Oh yes, actually when we check the function of microRNAs after the construction of model using the kind of microRNA target scan or something, and we try to estimate the function of these microRNAs. Actually sometimes we found similar genes were regulated in these panels.
One more. The last question.
Only
Speak
one
out.
question. Okay.
No.
Oh, sure. Sure. I want to ask Dr. Matsuzaki, for how long does it take from you take sample from patient and to microRNA result, or until the result is clear? How long does it take?
This is clinically really important thing. In the experiment, we do within the 3 days, but this is the clinical, not clinical setting. This is the laboratory setting. So to do more severe, more strict clinical test, I think we need 1 week or around, I think.
So for 13 kind of cancer, right?
Right. In future, we try to do that.
Okay, so because the time limited, we probably can. If you have any other question, you can ask later. Okay, so let's thanks again for Juntaro's talk. And 那大会有一个那个不学国语，你可以说吧。
我们要等一下，不知道什么时候
我们做一个大合照，好不好？那就坐在这个园子里，然后大家集中。